us in the audience, so I feel like I can get away with it. So.
All right, so I'm setting us up for the webinar. Okay, so I think we're ready to go. I'll go ahead and get the webinar started if you're ready, Bashira.
Yeah, I'm ready.
Okay. And we got folks hopping on. Let everybody get on.
Okay.
Welcome, everyone. We're just letting folks hop on. We'll get started here in just a few minutes. Okay, that looks pretty good. It looks like numbers are starting to settle there. So hello, everyone, and welcome to another Science Cafe with the Atlanta Botanical Gardens. We're super excited to have with us from the Auburn University, Bashira Chowdhury, who will be presenting on pollination puzzles in the South. Just a few things. Bashira will be speaking for approximately 30 to 45 minutes, somewhere in that vicinity. And then we'll go ahead and have a question and answer section after that, where she can answer some of your all's questions. And we do have a little Q&A feature there at the bottom of the screen that you can see. And if you have any questions, just go ahead and you can type those in there, and you can do that at any point, but we won't address those questions until the end of the talk. Also want to let everyone know that we are recording this. So if you had friends that weren't able to make it for some reason, this will be available on our website. And I can go ahead at the end of the talk and I'll share the address for the web address for our Science Cafe webpage, so folks can check that out. And yeah, just a little bit about Bashira. She is a researcher in pollination ecology at Auburn University with a specific interest in how knowledge about pollination can serve rare and threatened plants in the southeastern US. She works with Auburn University entomologists to study the ecology and conservation of Alabama's endangered Spigelia, or pink roots, along with two federally listed threatened pea plants, Apios priceana and Dahlia foliosa. So without further ado, I give you Bashira Chowdhury. Thank you, Bashira.
Hi, thanks, Ian. Thank you again for having me at today's Science Cafe. So we'll just go ahead and get started. So what's on tap for you guys is we're going to start off with a little bit of Pollination 101. It's hard to solve a puzzle about something that you might not know quite much about. And then we'll get into our puzzle about Spigelia. And then finally, the first question is actually going to come from my dad. But I think it's the question that a lot of folks are always thinking about when we're talking about science. So first off, pollination. Pollination is simply just how plants date and mate. That's it. There's not even a bee in the definition. This is not about bees. This is about plants. All pollination is pollen moving from the male part of a flower to the female part of a flower. That's it. That's at its core what pollination is. And I know for the botanists out there or the plant loving folks, I have made a plant look kind of weird, but this is how truly what pollination ecology reduces down to. Moving pollen from the male part of a flower to the female part of a flower. That's it. There are no bees involved, not even the letters. And really, to understand why, though, we tend to focus on pollination, we have to take a little bit of a step back, and we have to look a little bit more carefully in how plants reproduce. And plants reproduce two ways. And even though they seem unfamiliar to us, we actually do a little bit of this reproduction ourselves. So plants reproduce, the first way is what we call asexually. So, it seems like it's clonal, but it's not, which is why that wasn't a mistake on that petal right there. It is meant to be actually a pink petal. Asexual reproduction is not clonal. It's not exact copies. It's just something similar, but it's very similar. And asexual reproduction is something happening in your bodies every day. Your skin asexually reproduces, but it's not completely replicate. It's not a complete perfect. It's not completely perfect every time. And in fact, this is how cancers get started, through asexual reproduction. On the converse side, there is sexual reproduction. This is where pollination comes into play. Sexual reproduction is all about mixing stuff up. And in the mixing stuff up, you create all kinds of new combinations of things. That's how out of a white flower and a sort of kind of pink flower, you can sometimes get a pink, white, and possibly a little bit of red in there. That's the core of sexual reproduction. It's a game of combinations. And pollination is all about helping that game happen. And that's where pollination matters. It matters in how plants sexually produce themselves, how they sexually create kids. That's what really reproduction comes down to. How do you make kids? And it doesn't actually matter for all plants. It only matters for some plants. So it really, using some technical terms here, but it really just matters for plants that are what we call gymnosperms, probably more familiar to you as a pine tree. It's those conifers. And then, of course, angiosperms, which you're probably also familiar with because you eat them when you eat corn, when you eat tomatoes, when you eat an apple. And those are commonly known as the flowering plants. So this is where pollination matters for these two groups of plants. So now that you know for who it matters, let's get into how it matters. And really, this is the core of pollination ecology, and this is what I work on, is the how in this definition. And it's really broken down into two things: whose pollen it is and how that pollen's moving, or in a very crass way of saying it, who's your daddy and how'd your daddy get there? That's really what pollination ecologists study. And we're not, our puzzle today is not really going to focus so much on who's your daddy. The who's your daddy side of pollination ecology is amazing and cool. And it gets into, it does do the paternity testing that would make Jerry Springer blush. You would be surprised at how promiscuous plants really are. And it also gets into some mechanisms that are very common to us, because pollen and stigmas recognize each other in a manner that is no different in how our immune cells recognize self from non-self, otherwise known as the MHC receptor complex. But our story today is going to focus on how pollen moves. So today, your version of Pollination 101 is going to focus on how pollen moves to help you kind of understand this puzzle a little bit more. So pollen can move 4 different ways. It can move on its own. The flower can do it. Physically, this anther can touch its own stigma, or the wind can do it, or water can do it, or animals can do it. And usually we tend to focus on the animal pollinated plants because they get us interacting with some things that we like a lot. So for those in the South, they're going to be thinking a lot about their bees moving pollen, but it's not all a bee story. The South for certain is not all a bee story. You've got flies involved. you've got beetles involved, you've got hawk moths involved, and then you've got you've got hummingbirds involved, as depicted in this very realistic cartoon of a hummingbird. And so you've got a variety of animals involved, but we tend to focus on the bees. And I'm going to take a small little sort of side tangent here about bees. And we're not going to talk about bees after this. So realistically, the reason that you probably hear about bees and pollination is because bees spend their entire life around pollen. So from larva to adult. So when you see a bee out there on a flower, it's usually a female. It's usually a single mom. Most of the female bees that you see, the bees out, they're usually single moms. And they're out gathering food for their kids. So at the end of the day, this pollen is really food for their kids back at the nest. What they're doing is they're gathering this pollen from a variety of flowers. They're packing it into a little pollen ball. They're putting that pollen ball in their nest, and then they lay their egg on it and they seal it. And that's it. So realistically, these, bees are not out to be pollinating anything. Their care is not for the plant. They care about their kids. They are no different than your mom. Your mom wants to make sure you're fed. And that's all these female bees are doing. In the process, you can have some bees, some sloppy moms, basically, that as they're going from store to store, they might drop a few items along the way. And in that process, they can pollinate. But it doesn't necessarily mean they're all pollinating. And we tend to focus on bees simply because, in our part of the world, bees do a lot of the heavy lifting when it comes to pollinating as these female bees are shopping. But our story really isn't going to involve bees. But I just wanted to clarify that because it's really hard to not think about pollination and not think about bees. Now, given all that, which way do plants pick? And is there any right way that plants can be pollinated? And in all honesty, that answer depends on the plant. So all 4 ways that you can move pollen are completely valid. There's nothing wrong with them. All it comes down to is which of these ways helps you produce your kids. And that's a little something we call fitness that, in all honesty, should be its own Science Cafe talk. So now that you've had a little bit of Pollination 101, we can get into our story. So, like all stories, they start somewhere. So this story is actually going to start with Kathy Matthews. So I'm hoping Kathy is out there in the world, out there in our virtual Science Cafe here. I tried to make sure she was invited because this story truly starts with her. So Kathy Matthews, she's now a professor at Western Carolina University, but she was doing her PhD at UT Austin. She's working on Spigelia. And she is the world expert on this plant, by the way. And Kathy, if you're out there, feel free to chime in and make corrections as needed. But in many ways, I'm going to be presenting her part of the story first. So Kathy and her mentor, Lawrence Gilbert, found out that this plant started out in South America. So realistically, all things Spigelia start somewhere in this big black circle that I've circled. And it's ambiguous because it's hard to really pinpoint where anything starts. You know, these things started millions of years ago. So it's sort of like a little bit of historical research to figure this out, you know, and plants don't really keep written records from millions of years. So we've got to tease it out. And so she's been working with her mentor to tease this out for quite a while now. But our story really starts in South America. And then from South America, a while back, Spigelia did this. It traveled, it moved. And this is the interesting part of it. And no, I didn't make a mistake here in that it did actually cross somehow the Caribbean Ocean. And somehow it went through Central America into Mexico. And yes, they're meant to be 2 lines because it looks like it did this a couple times. And that's interesting. And And it gets even more interesting when you look at the south. And so this is to give you a sense of what Spigalia looks like. So when we're talking about Spigalia down in Mexico in the tropics, it literally looks like a hummingbird bar. This is a plant that is most likely hummingbird pollinated. And it's, a hummingbird is going to come through and just be taking little sips from each one of these flowers. And this is what Spigalia looks like in our neck of the woods. So I actually took this photo on Sunday. And this is Spigalia gentianoides down in southern Alabama, and it sort of still has that same kind of shape. But it actually, oddly enough, I know I said that I wasn't going to talk about bees. This actually happens to most likely be a bee bar. But either way, you can kind of tell, like, definitely similar shapes, but definitely took on slightly different forms as it traveled. And this is really what Spigalia looks like in the south. So there's this plant, this one member of this genus, this one member of this group of plants, Spigalia marylandica, that is in a lot of different places. And no, I did not, misprint all these different names. I tried to give you a sense that all the others are in smaller little patches across the southeast. And Spigalia alabinensis is actually really tiny. It's only found in one little spot in the middle of Alabama. But for the most part, this whole landscape is dominated by one major Spigalia and then a couple other scattered about. Another way to look at this, building further on what Kathy's done, is to look at the family tree, because a lot of things tend to travel as families. And so when it comes to sort of the evolutionary biology of it, we like to look at trees. Darwin taught us to think about evolution in trees. We all come from a family tree. We all come from a common ancestor. So the colors here signify sort of the different families. So the dominant family is the Marylandica group. And that's where you see the big red around Mississippi, Tennessee, that whole zone. That's all the Marylandica branch. And then you've got two little circles here that are actually related to each other. And so you've got one circle over in Florida, and then you got one circle over in Texas. And they're actually closely related to each other, despite the fact that you've got a big, huge, different family or different group of plants in the middle. So basically, this is what, this is the interesting aspect to how on earth did this happen? And, where does pollination fit in all of this? And that's where our puzzle begins. That's kind of where I came into this. So I was curious about this whole thing, and I had first learned about Kathy's work a couple of years ago. And so I thought, well, it would be fun to kind of figure this out a little bit, figure out what's going on. Like, how did this happen? How did something from the tropics make it in Alabama? How is it making it all the way to Kentucky? Like, how is it making it in, the United States? The tropics are really different than the United States. Like, it's a totally different world. You got different animals, different everything, different music, goodness gracious. So taking a closer look, this is I borrowed this from iNaturalist, which, for those of you not familiar with iNaturalist, is a website where those who love nature can share all the cool things they found in nature. And Spigalia marylandica definitely has an audience out there. And so there are a lot of sightings of Spigalia marylandica. And you can see it's kind of all over the place. It even made it all the way up to New York and Ohio, places where people don't sometimes really even want to go. And so when we take even a closer look, and I actually borrowed this from Kathy, she kindly scanned this in from her PhD dissertation. So this is what marylandica looked like in 1999. This is what it looks like now. So you can tell it has expanded. And granted, there are more dots in 2021. And granted, you've got, Kathy used original herbarium records, and this is people citing it. And so, of course, you've got slightly different, sampling techniques here going on, and sampling efforts. But the major point I want to point out is the big area in black. It wasn't there before. And it's going to places like Indiana. It's going to places like Ohio. It's going to Philly. It's going to New York, and that's interesting because it's over a very short period of time that it's spread. And again, Indiana is nothing like the tropics. I mean, they don't have plantains in Indiana unless you go to a grocery store. So realistically, there's something really interesting going on here. And also, when we look a little bit more carefully, it makes us wonder, like, well, how is it making it up there? How did something from South America make it in Indiana? And so that is the heart of our puzzle today. And we're taking a look at it from the pollination aspect. And as I mentioned, our story really focuses on the how, because that's the heart of what this project's about. But you can't do pollination ecology without doing your due diligence. You still got to look at who's the daddy. And without going into any complicated efforts into figuring out, there isn't any paternity testing done here. We're not going to be making any Marylandica flowers blush with all the different daddies that their seeds might have come from. But realistically, we did some simple tests just to figure that out. And we found out that it can mate with itself and others, meaning that basically pollen from the same flower can help set seed, or pollen from a different plant can help set seed. It doesn't actually matter where the pollen's coming from. And I put this photo here to show you that sometimes what seems like a very complex thing in science, like who's your daddy, like, oh my goodness, we need to bring in the DNA kits, can be really simple. And all that is we are actually physically moving the pollen, whether it's with a watercolor brush, or if we want to be more technical and quantitative, we will use a fishing line, because you can actually count the number of pollen grains that get stuck on the end of a fishing line. So I could tell you that there's exactly 60 pollen grains ending up on that stigma. But regardless, it's really straightforward. We bag it with bridal veil mesh. So just so you know, also, bridal veil isn't just for getting married. It is also for telling who's your daddy. For plants. But realistically, all it is us physically moving the pollen and preventing pollinators from getting to it. But there's a pretty simple way to figure that out. But now our story is going to move into how pollen moves, or how your daddy got there. And that's really the heart of what we wanted to figure out, because how your daddy gets anywhere is very different from the tropics to temperate areas. From a place like Guyana or Venezuela to a place like Alabama or Mississippi or Tennessee. So in Spigalia, and I know that the little cartoon on the screen, it looks like really bad Christmas ornaments, but that's how we view Spigalia. Sometimes all the fanciness really comes down to red cones. But there are a couple ways that Spigalia can reproduce, or pollen can move in Spigalia. One is that the flower could do it itself. And another way of saying that is just autopollinate, self-pollinate. Or pollen can move between flowers on the same plant, which is a fancy word called gaetonomy. And for those of you out there with folks studying for SATs, that could be a great SAT word. And pollen can move between plants, and that's called xenogamy, another great SAT word. But all it comes down to is there's 3 really different pathways where pollen can move. And we wanted to understand, well, how does it do it? And does it play any role in how these plants are making it far away from where their ancestral heritage is from? And so what we did is we physically went out and went to a variety of Spigalia marylandica populations from Florida all the way pretty darn close to the Tennessee-Kentucky border. And we examined this, we looked at this, we studied this. And to make a long story short, what we found is closer to Florida, you depended on hummingbirds. It was strongly what we call xenogamous, or you really, really needed your hummingbird to move that pollen. But as you inched northward, it actually started to become more reliant on itself, to the extent that flowers outside of Nashville and north are primarily autopollinated. And it doesn't make a flip of a difference if you've got an animal there. It actually is more, it actually produces more kids when it autopollinates than when you got an animal involved, which is interesting that it has become more self-reliant as it's gone up north. And of course, as scientists, we keep asking how. The key, in any scientific story, I'm sure for those of you that, out there in Science Cafe and out there in the cafe are thinking like science is all about asking how. So you keep asking how on earth did this happen? So we looked a little bit more closely at the how. And at the how was basically, Spigalia has an unusual way of presenting its pollen to pollinators, in that it does this thing called secondary pollen presentation. So using a part of the female reproductive structure, the style, it sticks the pollen right onto the style. And then as you creep up north, where it sticks that pollen gets closer and closer to the stigma, basically where pollen needs to be in order to help set seed. And the flower also gets shorter. So it's a shorter flower, a little bit wider, and pollen, it literally sticks that right onto that stigma. So it looks like the flower itself is facilitating its own ability to pollinate. And so, of course, you're asking, that's all good and well. And so this is where I know Ian wanted me to expand a little bit further, but I think that's really where our puzzle leaves off. And I'm actually going to start the Q&A with my dad, because I think at the end we hear all these really, really cool things in science. But my dad always asks the question that I'm sure all of you are thinking, what's the point? And by the way, that is actually my dad, and my sister actually took that photo. But the question is really at the heart is, what's the point? Why on earth study all this? Like, what's the point of understanding that Spigalia marilandica is autopollinated in Tennessee and it is pollinated by hummingbirds in Florida? And, from a practical standpoint, Spigalia, it does have some practical importance, particularly for those in the global South. So a lot of folks in the global South, they have helminth diseases. We don't have them up north. And Spigalia is a known anti-helminth. And so understanding how to keep Spigalia around, how to produce it and so forth is important from that aspect. But I think that sometimes in science, we spend so much time on the practical that we forget about that sometimes there's a fundamental aspect to all this. And that's really the point here, is that our world is changing really fast. And we need to work with our plants to solve a lot of problems. And it's not just about food. It's about everything. Plants support a whole lot of things in our lives, from food to our buildings. I'm sure all of you know that we're in a lumber shortage right now. And plants also create our environment. They're a part of what we appreciate about this world. And so we have to understand what happens to plants when they move. And it's no different than if you want to build a rocket to go to the moon. You have to understand gravity. If you want to cure cancer, you have to understand how cells divide. And if we want to solve all these big environmental challenges that involve plants, whether it's food, whether it's conservation, whether it's just simply we want to have some of this cool stuff around, we have to understand how plants move and what happens to them. And that's really the point of Spigalia. This is a project simply about understanding what happens to plants when they move. And so that's really what the point of Spigalia is what happens when a plant moves. And with that, that's that's our puzzle for the moment. So I'm happy to answer any other questions you might have about pollination. So I'm just going to read off. So Megan asked, how long ago did Spigalia end up in North America? And how quickly do you think that northern populations began to show this shorter self-fear adaptation? Awesome question, Megan. The big answer to that is I don't know. And so the basically when it comes to plants, unfortunately, plants don't speak our language and can't tell us exactly when. I can tell you that we can start looking when it comes to answering the very question you're asking. It's going to come down to a combination of looking at the molecules and looking at the fossils to understand. And Spigalia fossils are probably going to be hard and fast to find because it's the tropics and you don't have too many too many fossils from the tropics, but it's going to be some combination of teasing it out with the molecules and the fossils at that end. As far as becoming more shorter and selfier, basically it comes down to we're trying to tease that out. So this year, in this fall, we're actually going to be running an experiment where we're taking Florida Spigalia and planting it in Tennessee, and Tennessee Spigalia and planting it in Florida to first understand, is there a genetic component to any of this? So if Florida Spigalia, which totally needs animals, actually still needs animals in Tennessee, then we know that it is genetically based. And from there, we can start teasing it apart, again, using a lot of the work that Kathy's done to figure out how did this happen. I will tell you, in order for any of this to happen, that particular trait had to have already been in Spigalia. You can't just suddenly like have, things come out of nothing. So this trait had to most likely it has to be a part of the heritage of Spigalia. So hopefully, Megan, that answers your question. All right. And then I think I'm supposed to hand it over to Amanda to field questions.
Yeah, I guess Ian got disconnected. So I will shoot you the question if you want to answer.
Sure.
Yeah, I am back on. I'm sorry about that. Sorry about that, Bashira. I got just kicked off all of a sudden.
No. If it's easier, I can just read the screen to answer the questions.
If that works for you, that's great. Thank you so much. And
okay, so
sorry I got kicked off, but
that's okay. So Mark asked, does relying on self-pollination reduce the fitness of Spigalia? That's again, awesome question, because that is like sort of the heart of a lot of what folks like Ian do and Atlanta Botanical Garden do. They're worried about things like this. So the answer to that is I don't know, actually, because again, that relies on field tests that we're currently doing. So that's where you've got to plant seed that was created from self-pollen, or basically the plant being its own daddy, and from outcross pollen or from a different plant. I can tell you in general, it appears that it does not reduce fitness as far as seed set. So basically what we found so far is that pollen from yourself or pollen from someone else, from a different daddy, produces basically the same numbers of kids, relatively the same time of the year. So right now we just got to plant those seeds to see if those kids are just as fit. So hopefully, Mark, that answered your question. So for Donald, over the past 2 or 3 decades, Spigalia has increased in popularity and availability in the ornamental nursery trade. Any idea what the geographical source is for the plants being transported by the commercial horticultural firms? Super awesome question, Donald, because that's actually how I think Spigalia made it to New York, by the way. So it has entered the ornamental nursery trade is responsible for moving so many plants around and facilitating so many questions. So we don't know what the original geographic source is. I can tell you that will be actually some DNA testing at the end of the day to figure that out. But usually what we found is that a lot of native plant societies tend to bring this into their state. So for example, like in North Carolina, for folks out in North Carolina, it looks like Spigelia entered into some areas of North Carolina where it's never been, courtesy of native plant sales. So that's, that's kind of where it starts, and it kind of moves on from there. And of course, Spigelia has definitely captured sort of that imagination because it's a great hummingbird plant. As you can tell, in many ways, it's like a hummingbird bar. So it has sort of entered the trade from there. But long story short, Donald, we don't quite know. But I can tell you that most likely all those spottings you saw on iNaturalist, those are all hort trade. That's what Kathy and I were talking about a couple months ago, that it's most likely hort trade. So Rachel's question is, do other plants show signs of becoming more self-reliant? Yes, they do this all the time. You eat them. You eat them all the time. In fact, our food system would collapse if we didn't have plants that were self-reliant. So things like tomatoes are self-reliant. They're largely selfing. I know a lot of people think that bees are the reason that they have these wonderful juicy tomatoes. No, you can thank a crop breeder. Realistically, we've bred our crops to be largely selfing. So a lot of things are selfing. And in general, there's sort of a rule of thumb in ecology that you tend to see plants become more self-reliant the further they are away from sort of their optimal environment or their diversity center, or things that basically facilitate their pollen movement. And we've seen this classically most recently in cities. We've seen a lot of plants that have moved into cities, particularly weedy plants, actually become more self-reliant. So there's actually a really cool study out of France where they were looking at something that is no different than our dandelion, and found that it has essentially become more self-reliant as it has entered French cities. So hopefully that kind of addresses your question there. And then for Heather, I've planted Spigelia in my yard. Do you think plantings played a role in the recent northern movement? I don't doubt I strongly would hypothesize that plantings did. I don't necessarily think it's a bad thing. We have been moving plants for ages. In fact, if we didn't move plants, again, we wouldn't be able to eat. So the best story about moving plants that I can tell you is corn. Corn started out as this little tiny weedy little grass in southern Mexico. It's a tropical plant. Corn is tropical. A lot of people don't know that. And it crossed deserts. It crossed Texas. And if you live in Texas, you know how hard it is to cross Texas. And it went through the Great Plains. It went to Michigan. And I'm sorry for those of you from Michigan. I'm sorry you're from Michigan. But it made it to Michigan. And to do all of that, people have been moving. So people have been moving plants. And so when it comes to moving plants, we've been doing it the whole time. We've been facilitating how plants move for ages. And we couldn't live our lives without moving plants. I will say this Science Cafe is sponsored by the Atlanta Botanical Garden. So I'm sure I have to give a very cautious, like, don't move plants all over the place, because we do have invasive plants. We have things like privet that I'm sure the Atlanta Botanical Garden does not like. We have things like mimosa. We have things like kudzu, and so forth. So there are plants that we don't want to move, but there are plants that we have been moving for ages. So, but a very long answer to your very short question is that probably yes, that is how Spigelia made it to Indiana. So most likely it was plantings. From someone anonymous, is it possible that hummingbirds that pollinate Spigelia in the South do not range further north so that self-pollination is more common? That is possible. I also think that there's possibly, we can't just say that when it comes to all these traits that you see, that yes, pollinators help select some of them. So that famous case that I'm certain that some of y'all have heard out there of Darwin's hawk moth and his flower, that's out there. That is true. Like definitely pollinators like hummingbirds help to guide some of these floral traits, help to select is for the evolutionary ecologists out there, the correct technical term. But it's not the whole story behind these flowers. So we can't just say it's only the hummingbirds. There's possibly the role of climate, because flower size is very much dependent on how much stuff you got in your soil, how much access to nutrients you have, how long your season is. Remember, it gets cold up in Tennessee. Those Great Smoky Mountains get snow, and the Spigalia is up there. And Florida doesn't get snow. You got longer growing seasons. So there's a variety of factors. So there's possibly no one factor that can really explain why it's self-pollinating. But that's, again, that gives folks like me a job. We're supposed to go figure that out. So again, we can keep adding to the fundamentals and keep adding to more ways that we can make our world better. For Vicky out there, I love the phrase, there is no bee in pollination, to explain self-pollination. Thank you. Yes, 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 yes. If you get anything out of all my craziness today, remember, pollination is all about how plants date and mate. I am just really, I study plant tinder. That's all I do. That's really what I do. And if you become a pollination ecologist, don't tell your parents that, because that's not what they want to hear. But that's really what pollination is. It's studying plant tinder. And you always have to think it's about the plant first. And so then other things come into play. And then Megan again, do you see lots of morphological differences in flowers and pollination style and other Spigalia species? Yes. If you want to have fun and you just want to, if you just love plants, go to iNaturalist and type in Spigalia. You will see all kinds of crazy ways and crazy forms. And I just look at this as, particularly in the tropics, and I can say this for the most part in the tropics, a lot of things in the tropics are very much dependent on animal pollination. The tropics is an animal world. I know earlier I said that, there's 4 ways that plants can move their pollen. That's still true in the tropics. It's just that animal pollination dominates. Even things that are wind pollinated up here in, the South are actually really animal pollinated in the tropics. And so animals do all kinds of crazy, they select for all kinds of crazy things in plants. And if you want to see some crazy Spigalia, just look up the tropical Spigalia. It is literally like all different kinds of animal bars. And I apologize out there in Science Cafe, because I'm sure there are children out there. And so it's basically all kinds of different cafes that are being offered to animals as food. Because remember, flowers are just basically feeding troughs for animals. And when it comes to animal pollination, all these animals are doing is coming in and feeding, and plants are just hitching a ride. They're just sticking their pollen on those animals to get it to move. So I hope y'all enjoyed learning a little bit about Spigalia and kind of entering a little bit of the world of pollination ecology. And feel free to reach out if you've got any more questions about pollination. And thank you again, Ian and Atlanta Botanical Garden, for inviting me.
Thank you so much, Bashira. And I apologize for getting cut off there for a little bit, but sometimes that's the way things go with Zoom. So, but yes, thank you again. And yeah, for such a wonderfully engaging talk. It was quite wonderful. I actually do want to go ahead and just share the screen,
Okay.
my screen. And I've just got a slide up. Can folks see that now? Or no, actually, let me get out of there. Let's see. Share screen. Here we go. Now it should be, there we go. Are you seeing it now? Huh. Do you see that screen? Can folks see that now, Bashira?
Yes.
Okay, great, cool. So yeah, I was saying that we are recording this event, so folks can go ahead and they can click that link, and that is our Science Cafe website with the Conservation and Research Department at the Atlanta Botanical Gardens, and they can go ahead and see all the other talks that we've done in the past, and Bashira's talk will be up on there, and we'll try to have it up on there by the end of the week next week at the latest. And again, thanks everyone who came out tonight. You know, we couldn't do it without y'all coming in, so it's wonderful to have everyone here. And we are going to have on July 15th, as always, the third Thursday of the month, we're going to have a talk with Dr. Caitlin Kahn, who's a biologist and assistant professor at Berry College. And she's going to be speaking on the broomrape family, or Orobanchaceae. And that's quite well known for their parasitic plants. And she does a lot of genetic research into that family. So we're really looking forward to having her next month. And also, if anybody has any questions or suggestions, feel free to reach out to me at isabo@atlantabg.org. And as always, we have a Instagram account at Atlanta BG Conservation. So feel free to check that out and follow us if you're into that kind of thing. And thanks everyone for coming out. Have a great evening.
Thank you.
Thank you. I'm going to go ahead and end the, okay, I'm not going to end the meeting, but I'll go ahead and stop the recording.